So welcome to the Brain Imaging Research Center, or BIRC Speaker Series at the University of Connecticut. I am the host, Fumiko Haifch, Director of BIRC and Professor of Psychological Sciences. Over the past 3 years, we've hosted a number of speakers. As you can see, some of the speakers over here on this slide here, Damien Fair, Jim Haxby, Tor Wager, Sue Whitfield-Gabrielli, and so on. And so thank you very much for those who's joined in the past and or who are joining for the first time and making this successful. You can please let your colleagues know that you can sign up to receive information about recordings and future talks by emailing birk@uconn.edu. So some housekeeping items. This series will be shifting to Friday afternoons at 1:30 p.m. Eastern Time, starting in December. And we did a little poll and we decided that that might be the more optimal time. And it seems to overlap a lot with other faculty meetings as well as some lab meetings of the various people that we've asked. Our next speaker is Professor Jason Yeatman from Stanford, an expert in diffusion imaging, and he's made a number of tools that many of you may be using already. This is in December, and in January we have Professor Roy Hamilton from University of Penn. He's an expert in transcranial magnetic stimulation, TMS. So as for this talk, if you have any questions, please put them in the chat box. As a default, we'd like to take it towards the end, but if it's something urgent, you can message us and we will try to accommodate, but generally we will take it towards the end. And let's see. I unfortunately have to leave a couple minutes early, possibly if it goes over, but the Associate Director, Professor Roland Hancock, will take over towards the end of the talk in that case. So let's get started. Today it's a great honor to have Professor Swathi Kiran as our speaker today. She is the James and Cecilia Ying Professor of Neurorehab in the Department of Speech and Hearing Sciences and Associate Dean for Research at Sargent College of Health and Rehabilitation Sciences, and Director for Strategic Planning at the Office of the Provost at Boston University. It's amazing that you can continue to line your research and these heavy administrative duties. That's quite impressive. She's received her PhD in 2020 from Northwestern University in speech language pathology, and she's an exceptional scholar where she tackles many very challenging topics in the area of aphasia. So for example, her research interests focus around lexical semantic treatment for individuals with aphasia, bilingual aphasia, and neuroimaging of brain plasticity following a stroke. She has over 120 peer-reviewed papers in these fields and has been funded by NIH and ASH Foundation. She is the co-founder and scientific advisor for Constant Therapy Health, a software platform for rehabilitation tools after brain injury, and is a fellow of the American Speech, Language, and Hearing Association. We are very thrilled to hear your talk today, Swathi, and please join me in welcoming Professor Swathi Kiran.
Thank you, Fumiko. I'm really happy to be here. I think I mentioned that I've been attending many of your talks this past year, and they've been amazing topics. So thanks for having me part of this group. Let me go ahead and share my screen. So hopefully you can see my screen.
Yes, perfect.
Okay. So I need to get a few of my disclosures out. You've already mentioned them, but I should say that I have an ownership stock for Constant Therapy Health, and I have been funded from NIDCD for most of the work I'll be talking about today. Okay, so in the future, I envision that a survivor of brain injury will come to a lab or a clinic, and we will get a complete language or neurological assessment done. We'll also try to get a complete social profile, so asking them what their education is, what their occupation is, and what sort of goals they want in life. Then we'll use this information to predict that particular person's recovery profile trajectory. Based on this information, we will be able to make optimal prescriptions about whether that patient should receive individual intensive therapy, or they should receive some individual therapy or in social groups, or they should participate in educational or vocational training. So based on each individual patient's background and profile and relevant information, we'll be able to give this optimal prescription. But we're not there yet today. So why is that? The reason we're not there yet is because it's a very complex problem to solve. Acquired brain injury is a term that includes someone who has either had a stroke or a traumatic brain injury and hampers people from communicating and successfully participating in their life. In fact, it's one of the most debilitating chronic diseases that somebody can have. There are many factors that determine the degree to which someone with an ABI can recover their function. Among these are biological factors such as the degree to which physiological neuroplasticity, such as changes in dendritic spines and connections, occur. Also, specific individual factors such as the degree to which an initial severity, age, and lesion size and location influence the recovery also matter. And finally, the behavioral interventions and treatments that somebody can receive or get, including the salience and specificity, also determine the degree of language recovery. Now, our work at Boston University has focused on teasing apart these factors. I'm going to lump them into 3 buckets, but I'll mostly talk only about one of them today. We're examining brain structures and function that determine the degree to which brain damage influences recovery. Second, we've spent several years examining the nature of language and cognitive impairment that influence the inability to function successfully in an environment. And finally, we've also spent many years developing effective treatments for specific language and cognitive function deficits. But more recently, we've been trying to integrate all of these multidimensional factors to develop machine learning and computational algorithms to best predict which of these factors are important in recovery and how to personalize the specific kind of treatment outcomes. Now, today's talk will focus really on the neural markers because it's the BIRC group. So I'm hoping this will be of interest to you. And when I do this, I'm going to sort of talk about our work that has looked at lesion information, including gray and white matter structure integrity, diffusion-based white matter integrity, task-based fMRI and resting state fMRI, and the corresponding functional connectivity and graph-based measures that look at this. We've also been looking recently at fMIRS in these patients, and also something that I'm going to call overall brain health in this process. So what we're trying to do here is understand the influence of all these factors I just showed you to predict impairment and recovery. And just to bring everybody on the same page, the dashed green line on the top here shows you healthy controls and what they look like. And the color here is sort of the ability of language function. So when somebody has a stroke, right after the stroke, as you can imagine, there's a quite severe decrease in language functions. But as they recover over time, there's an improvement of language functions that happens. And this tries to get closer and closer to more normal-like behavior. But when they reach the chronic stage, they're still presented with language impairment, and some more than others. So some patients have more typical language functions, as you can see in the yellow line, they're trying to get closer to normal, while others have more severe language impairments, like in the orange line here. And many of these patients receive treatment, especially if it's introduced in the chronic stage, like what I'll talk about. And some of these, patients improve more than others. And what we see regularly is that there's a tremendous amount of individual variability. Some patients respond to treatment while others do not respond to treatment. And in the pursuit of the personalized rehabilitation work we're trying to do, the first step is to understand why some patients respond to treatment and why others do not respond to the same kind of treatment. So for the purpose of the talk today, I'm going to focus our attention on, what we've done so far and what measures we're going to be looking at. So in this case, I'm going to focus on impairment, which is sort of the language ability that somebody presents with at the chronic stage. And I will also talk about treatment outcomes and sort of classify them into responders and non-responders. To the extent possible, I will talk about when we're looking at both of these factors versus when we're only looking at one of these these measures. Also, I will give you some background about the several studies that I'm going to discuss today. The data draw from a fairly large-scale project that we recently completed. It was a large-scale multi-site project on the Center for Neurobiology of Language Recovery that was funded by NIDCD. And as part of our own sub-project here, we studied 30 patients with chronic aphasia due to left hemisphere. Of these, 26 patients were enrolled in a treatment group and 10 were sort of untreated controls, but it's important to note that 6 of them ended up also getting treatment once they finished the controlled phase. The other important thing is all of these are chronic aphasic patients. They really had this for a while now. As you can see, the average months post-stroke, and the lesion distribution show the kinds of lesions these individuals have had. Now, what happened to these patients in the study design is they would first come to us and we would get a complete baseline assessment done. So this is what I was saying, a cognitive linguistic profile assessment. And then we would also have them participate in a task and fMRI-based scan. We would also get a structural T1 scan and diffusion-based scans. Then they would receive therapy. And if they're in the treatment group, they would get therapy twice a week for 2 hours each week, up to 12 weeks, or when they got to 90% accuracy. If they were in the control group, they would sort of not get that intervention. And then after that, we would again do all the imaging measures again to look for changes. And then we would essentially do all the cognitive measures again. For the most part today, I'm mostly going to talk about this part of the pre-treatment assessments because we're looking for biomarkers of recovery here. Another quick overview of the kind of treatment we talk about, because I won't get to talk about this again in the talk. Here we're trying to help patients name or recover their naming ability. So they'd be asked to name pictures like hat. They go through the attempt to name it, then they have to sort the different category pictures, then they review the semantic attributes that have to do with hat from other distractors, and then they attempt to name the words in different contexts. What we do here is calculate the effect size for each participant as a function of this treatment, and we also calculate something called the proportional maximal gain, which takes into account how many items they get correct at the baseline. So we're really trying to calculate how much they've improved or gained from that intervention. Okay, so let me go through all our findings for some of these potential markers that we've studied. In one of the first studies, we looked at lesion information and the extent to which gray and white matter structural integrity predicted outcomes. This study had 30 patients. And in the first analysis, we essentially just broke down gray matter integrity in terms of percent lesions spared in sort of 5 cortical masks. We also looked at white matter integrity that was indexed by spared tissue and fractional anisotropy from 4 homologous left and right tracks. And then we combine them together to create something called a gray plus white matter measure, which essentially uses a combination of those two. And we compared whether gray matter only was better than gray matter plus white matter information. And the linear regression models here reveal that both gray and gray plus white matter clustering predicted baseline aphasia severity and naming skills, but only the gray plus white matter clustering predicted treatment outcomes. And I think that's sort of an important point that I'll make throughout. We also found that Spearman correlations with that showed that when we did not control for that overall lesion volume, we found that these measures were related to language, but once we adjusted for lesion volume, these essentially went away. So how much brain you have remaining sort of predicts how well you can perform on a particular language task. But when we looked at specific FA measures, we found that there were 2 ventral left hemisphere tracks that were related to naming performance and treatment, independent of lesion size. So lesion volume and gray matter measures were sufficient predictors of overall aphasia severity, but left IFOF and left ILF actually added predictive value to naming outcomes. So that's that proportional maximal gain that I was telling you about before. So diffusion metrics that show white matter tract integrity will add more power, predictive power to recovering to language recovery. In a more recent study, we sort of looked at this a bit more carefully. We examined diffusion-based white matter integrity in the left hemisphere, and this was really hard to do because we had to take care to delineate tracks in the damaged hemisphere in these patients, and we used automated fiber quantification, which is what you'll hear next month, I suppose, where you can sort of see the renderings, in this case, of sample tracks from one patient along 3 tracks, the left arcuate fasciculus, the left superior longitudinal fasciculus, and the left ILF. And what we found here, again, here we looked at proportional maximal gain. And what we found was that left arcuate fasciculus, left superior longitudinal fasciculus, and left ILF integrity were all associated with aphasia severity, the extent of your language impairment. But only left ILF integrity was associated with both aphasia severity and naming and the treatment response. So taking a closer look at this, baseline fractional anisotropy in the left ILF correlated with baseline aphasia severity. So this is looking at the tract on average, but when we break it down even further, looking at it in terms of the way AFQ allows you to look at it, baseline mean diffuse connectivity at the microstructural level correlated with treatment response in the posterior part of the tract. Again, this is suggesting that these left hemisphere tracts and the residual integrity is quite important in predicting treatment outcomes. In the next study, we took a slightly different approach, but this was a slightly expanded approach to understand if it was just sort of where the damage is or if there were distal effects to the damage as well. And we're calling this structural disconnection work. In this case, we had 76 patients with post-stroke aphasia who went through the same sort of language assessments. And what we were trying to do here was calculate voxel-based disconnectome symptom mapping. And what we do here is we essentially calculate the correlations between disconnectome maps. As you can see in this figure, they represent the probability of disconnection in each white matter voxel and correlate that to behavior. And then we compare this VDSM approach with BLSM approach, the usual voxel-based symptom lesion mapping, but we're doing this also in the white matter. And what we found was that there's a common core of white matter perisylvian network that's disconnected in about 90% of the patients with aphasia. And that's what's damaged, and I don't have access to that. But surrounding this core disconnectome, structural disconnections in the left temporoparietal network were significantly related to syntactic comprehension, syntactic production, and repetition. So this is beyond where the lesion is. And these language impairments are explained by disconnections that overlap, but they more importantly extend beyond the white matter lesion sites associated with each impairment. And the effects were strongest for syntactic comprehension and weakest for naming. But what these results show is that simple MRI, T1 and diffusion scans can provide a lot more information about not just where the damage is, but the sort of spread out effects of the stroke, and may also predict the degree of impairment and recovery. So far, just to give you a roadmap, I'm still sort of at the bottom of the slide here talking about lesion information and gray matter and white matter connectivity. But now let me go through some of the more fMRI studies that we've been doing in this case. So the first study is a simple fMRI task using picture naming that was used to identify 16 regions of interest. We studied this in 26 patients with chronic aphasia, and they all got the therapy I talked to you about. There's 10 control patients who did not receive treatment and 17 healthy controls who were also scanned. What we found here was that this naming therapy resulted in a significant increase in cortical activation. So as you can see here, post-treatment relative to pre-treatment, there's an increased activation in both the left and right hemisphere. But when we look at this more closely, because this is the group average, and across these 16 ROIs that we're looking at, it's clear that what's going on in the left hemisphere is sort of a reduction of normalization of activation. And what's going on in the right hemisphere is actually an increase in activation. And so we took this data from the left hemisphere and studied that even more carefully and found that the degree to which each of these left hemisphere residual regions were spared determined the extent to which there was upregulation or change of activation in these regions. So what we took away from this study was that there is an upregulation of traditional language areas and their right hemisphere homologs. Clearly there's activation changes happening in the left hemisphere, but there's a nuanced relationship between how much tissue is remaining in the left hemisphere and how much it can engage in the service of language recovery. So in the next study, this is not a treatment study, but we use dynamic causal modeling during picture naming to investigate the differences between aphasic patients and healthy controls. So this is just looking at what aphasia impairment looks like. And here we're looking at, in this particular case, we were only looking at 3 regions: the middle frontal gyrus, the left inferior frontal gyrus, and the middle temporal gyrus. And one thing about dynamic causal modeling is that you sort of have to set up all these possible model connections between these regions and estimate what the influence of one region might be on the other regions. This study was an early pilot study. It only included 13 patients with aphasia. But what we found was that in healthy controls, I said this is a picture naming task. What we found was that in healthy controls, as we would have expected, the driving input to the left IFG best fit the control data. But in the patients, it was the driving input to the left middle frontal gyrus that best fit the patient data. And what was also interesting was positive correlations between spared tissue and behavior and task effects to regions were also significant. So what that really meant was the more the patients with aphasia resemble controls in terms of their cortical integrity, the more MFG was inhibiting left IFG and left MTG. So this was our first foray into this finding that it's not just language regions that are important in the service of language function in post-stroke aphasic patients. Even regions like the MFG, which are not traditionally involved in language, might also help in the service of language recovery. So we sort of took this one step further with more patients, the same regions, MFG, IFG, and MTG, and we looked at a slightly different task in this case. It was a semantic feature task. But here, Erin looked at 25 patients and controls. What we found here, that was too fast. What we found here was, again, in both the control and the patient group, there's a heavy reliance on middle frontal gyrus. That's what you see on the top here with those high correlations, coefficient values. So even in the controls and definitely in the patients, there's a driving influence of the MFG onto LIFG and MTG. But what's also interesting is that accuracy on the feature judgment task was significantly related to the spared tissue in LIFG and MFG, but not LMTG. And the higher the spared tissue in these regions, the higher the strength of connection. So what this is essentially saying is that, as I just said, higher task accuracy is related to MTG, LIFG tri, but MFG is sort of modulating this relationship between LIFG and MTG in a way that really enables accurate task performance. So there's a dynamic relationship that's going on between these regions. In the next study, this was an even broader examination. This is all work done by Erin Meyer, who's now an assistant professor. So here we said, okay, we've looked at 3 regions. What if we expand this to more regions in the brain and really introduce not just DCM connectivity, effective connectivity, but really introduce lesion information into this mix. So this is a complex study, and I'll sort of break it down a bit. The first thing we did, unlike the previous 2 studies, was we looked at many more regions in the brain, and we looked at bilateral connections in the brain. And we set up 4 sets of models. The first one is a no damage model, so that's what healthy people should look like. But the remaining models are possible scenarios of what might happen. So in the first model, you have left lateralized connectivity when you have very little damage and posterior damage, or you have left lateralized connectivity when you have anterior damage, or you have greater damage in the left hemisphere, and now there's a shift happening to the right hemisphere, or there's complete damage in the left hemisphere, and really the shift, all the activation is happening in the right hemisphere. So using dynamic causal modeling, we tested these different models. The patients were 30 patients, and they're all doing a semantic feature judgment task. What we found was, while the healthy controls demonstrated a preference for the left lateralized connected models, that makes sense. That's what healthy people do. The patients actually split between some of them showing this left lateralized, and that was obviously related to their severity, but many of them showed this bilateral sort of connections, especially modulated within hemisphere for both left hemisphere, as well as those connections in the right hemisphere. But that was just part of the study, because we really wanted to bring in the lesion information as well, because we thought where the connectivity ends up will determine where the lesion was in the brain. So in this case, we looked at the interaction between lesion site and profiles of functional connectivity, and I won't get into the details because this is a fairly extensive PCA regression analysis. But I'll point to 2 important things in this slide. Within the patient group, what we found was damage to the left superior frontal structures resulted in greater right hemisphere connectivity. That's what I'm showing you here in pink. So there's damage to the left hemisphere, there's a shift to the right hemisphere, whereas damage to left ventral structures resulted in heightened modulation of the left frontal regions here, and that's what you see in yellow. And that's sort of exactly what we were predicting might happen here. But again, the take-home message here is that depending on where the lesion is, the connectivity shifts in the brain to either frontal or to the right hemisphere, depending on how large the damage is. I think that's an important point to make, because it's not just left hemisphere versus right hemisphere. It sort of depends on where the lesion in the brain is. And the more important point here is that connectivity shifts to left and right interhemispheric connections if there's just not enough tissue in the left hemisphere. But if there is, that's really what's driving the activation. So I'm going to switch over. I was talking about effective connectivity using DCM. I'm going to switch over and talk a little bit about a different kind of functional connectivity. Here we're just looking at connectivity between regions using task-based fMRI. So here we also studied about patients who received treatment. There were 20 patients. And there were 10 additional untreated patients and healthy controls. And they're all doing a naming task here, and they're receiving this therapy I talked about earlier. Now, all of these patients were first compared to healthy controls in terms of identifying a naming network that was different for them. So everything you see, all these blue connections you see here are patients who have an abnormal connection relative to healthy controls. So all of these 31 connections are abnormally low in patients relative to controls. Then we looked at these abnormal connections more closely and used linear mixed effects models to understand if these are different in patients who are trained versus untrained, and if they change. So as I said, we had patients who were treated and who were untreated, but we also had within this group of treated patients responders and non-responders to the treatment. So this graph on the right sort of shows three groups. The blue line are the responders to the treatment, and you can see that they actually show a change in the functional connectivity of these regions in a way that the connectivity improves over time. The middle line is the untrained group. These are folks that never received treatment and should not have changed as a function of this connectivity, should not show any changes in connectivity. And the lower group here in orange are actually the non-responders, and that's interesting because they actually show a decrease in their functional connectivity as a function of time. So what we know here is functional connectivity increased significantly from pre to post-treatment in patients who benefited the most from naming therapy, and not the untreated patients and not the non-responders. And so it does look like responding to a naming therapy is associated with increased connectivity that are functionally abnormal before this treatment. So the same work, this is done by Jeff Johnson as part of his dissertation. So he also then looked at graph measures that utilize the sort of task-based connectivity, 26 patients all getting treatment, and now we're looking at a different task because we just want to look at the different patterns. And here, what we're doing is looking at graph measures of connectivity. So we are looking at degree of the network, and degree is a binarized value, and then we also look at strength. It tells you the strength between any two connections. We look at clustering coefficient, but I won't show that data today, and that's a measure of the tendency of nodes to cluster together. We also looked at global efficiency, which is a measure of the efficiency of communication between each possible pairs in a network. So what we did in this study was we looked at this global and local measures in the pre-treatment data. So we took these patients' brains before they received treatment and tried to see if there was something different about them. And indeed, there were the responders and non-responders, when separated into two groups, showed very different profiles. What you see here are differences between the responders and non-responders in terms of their node strength, global efficiency, and local efficiency. And what we reported here is that patients who had higher network strength and higher global efficiency improved more in naming therapy than those that had lower strength and global efficiency. And the important thing that I said, and I want to reiterate here, is that these are their brain scans before they receive treatment. So there's something about these patients who are likely to benefit from treatment that's already different before they receive the treatment than the patients who are not likely to benefit from treatment. So this relative preservation of efficiency may be a biomarker of treatment recovery, is a good potential. Okay, finally, we've also examined graph theoretical measures using resting state fMRI. So it's the same kind of paradigm, except now we're not looking at task-based data. We're looking at resting state fMRI data. They're still receiving treatment. And here we're looking at several networks. We're looking at the language network and the default mode network, the dorsal attention network, and the salience network. Again, we're looking at degree and strength, which is 2 measures. We're also looking at clustering coefficient here, is the measure of the tendency of the nodes to cluster together. And we're looking at global efficiency, which is a measure of communication between each possible pair in the network. Now, what you see here in this table is sort of relationships, interaction effects between session, that's time out of improvement in treatment, and specific graph measures. And if you see something that's going up in red, it means that the higher the graph measure, the greater the improvement with treatment. If you see something in blue that's going down, then it means that the higher the graph measure, the less the improvement in treatment. And what's surprising but really interesting about this is that in the language network, greater connectivity, the greater degree, is not necessarily associated with better treatment response, but greater efficiency and greater clustering is associated with greater treatment response. In the salience network and the dorsal attention network, actually everything is associated with a greater treatment response. So we have to look at this in a bit more detail. And what we found here was in the language network, patients who had higher average degree improved less over time, and patients who have lower average degree improve more over time. This is really head scratching, and I'll try to get to it a bit later. But in the salience network, it's sort of what we would expect. Higher degree improve more, and patients with lower degree improve less. What's happening in the language network is we're looking at something called degree. That's actually a binarized network, and many of these patients actually have a lot of damage in these regions. And that seems to be a big factor into how well they can use those damaged regions or not use them in the service of language recovery. So the takeaway from this study, which is in prep right now, is that global efficiency and clustering in the language network are positively associated with treatment response. And that's exactly what Jeff found, and Isaac found the same thing with resting state data. But organization and efficiency are more important for treatment response than the overall connectivity. It's not so much where the connection is, it's really how efficient that connection is that's going to drive the extent of treatment recovery. And this is especially true in the language network, where there's a lot of damaged tissue and we have to sort of control for that. Okay, so I said a lot of studies. Let me try to sort of bring it all back together a bit. I'm going to present the same model I showed you earlier, but now with a framework, or a framework of what we think might be going on in terms of recovery in post-stroke aphasia. And here again, just to reorient you, the green line on the top here is what healthy controls look like. And this is sort of the trajectory of recovery from acute to subacute to chronic. And on the left-hand side now, I've added something called the functional connectivity or network topology in the brain. And what we think is going on here, and we've proposed this in this paper, is that normal optimal levels, like healthy controls levels of connectivity and language function, is what you see at the top here. That's what normal looks like. Now, when you have a stroke, at least in the acute stage, behavioral deficits coincide with abnormal functional connectivity and network topology. So the network becomes completely different from healthy controls, and it's completely abnormal. But in the following months, there is recovery and restoration of connectivity in the networks coincide with the degree of how much recovery there is. So the more they recover, the more normal-like the network becomes. Obviously, there are individual differences here. There are some people who recover more, as indicated by the yellow line, and others who recover less. Now, when you introduce treatment, patients whose network properties are already higher, they already had something going for them at this point because they recovered, their brains recovered more, are likely to be the ones that get more out of treatment. Whereas the patients who sort of did not show the same kind of reorganization of function don't get as much out of treatment. That's why we end up with the kind of responders and non-responders we do. But I think the most important thing here is that likely responders, people who are likely to end up looking like this blue line here, can be distinguished from the non-responders prior to treatment, right here, prior to treatment, because of their differences in the degree of network organization. So pre-treatment network differences are potentially meaningful predictors of treatment outcome. And what we're thinking is that people who have more efficient, better network topology after the stroke are likely to show more improvement because their brains can reorganize as a function of the intervention than people who don't have this reorganization going for them. So that's our current theory. And we've been trying to figure out if we can add to that, or, so that's what else can we make, can we add to this model? And in this context, we've done some work. This is work done by Maria Varkanitsa, looking at cerebral small vessel disease. This is again 30 patients. And in this work, what we do is we look at all the other parts of the brain. We look at white matter hyperintensities. We look at enlarged perivascular spaces. We look at lacunae and global cortical atrophy, and mark out what's going on in the brain outside of the stroke. And then we calculate the stroke lesion volume as a factor. We look at aphasia severity because we know that these two are highly correlated. And we look at demographic factors as well. And what this analysis has shown is that the severity of small vessel disease is strongly associated with the treatment-induced aphasia recovery in these survivors. Patients who have less severe cerebral small vessel disease are more likely to exhibit greater treatment outcomes than those who have more severe disease. And importantly, this association is above and beyond aphasia severity at the baseline and above and beyond stroke lesion volume. This is what else is going on in the brain. And we also found a negative association between the severity of CSVD and nonverbal executive function. So patients who have less severe CSVD are more likely to perform better in executive function tasks than people who have more severe SVD. So this sort of leads us to a theory that we're thinking that severe CSVD may lead to poor language therapy outcomes by decreasing the neural and cognitive resources that are essential for recovery. They just don't have the same kind of real estate to engage in language therapy. And we think that CSVD, which is really a marker of brain reserve, and it's a robust factor for post-stroke dementia, even in stroke patients, has this prognostic factor in terms of predicting treatment outcomes. So we might revise our model a bit by saying it's not just the degree of network reorganization, but the overall brain health of what else is going on in the brain might also be a factor in thinking about what predicts recovery in these folks. Again, this is work that we're still doing right now. Something else that we've tried to do is to look at other markers of language recovery, because we can only do so much with fMRI. And here we've started looking at fNIRS because it gives us a bit more flexibility in terms of the behavioral design and paradigms we can study. And this is very early work. This paper just came out this year. We were trying to look at healthy young adults and older adults and look at a very small sample of post-stroke aphasia patients. We're looking at exactly the same brain regions we would be doing in an fMRI task. So our probes are set up in bilateral temporal parietal frontal regions. And we're doing two different tasks with these people. We're having them do the same semantic feature task that we have very robust activation in fMRI. What we've found so far, at least, is healthy young adults and older adults show very clear left hemisphere activation, whereas the post-stroke patients don't show the same kind of activation in the left hemisphere. We've also looked at a domain general task, a cognitive task that involves arithmetic. Again, there's a lot of fMRI data showing activation for this task. And like the fMRI data, we find that the young adults and the healthy older adults show bilateral middle frontal and SMG activation when they're solving the harder problems. But the stroke patients are sort of getting there, but they're not showing the same strength of activation in this case. So this is very early work, but it's giving us another approach, another imaging approach to start studying the brain and hopefully get into more realistic fMRI behavioral tasks than we can do in fMRI. So that was a quick tour of all the work we've been doing to identify neural markers that predict recovery. I will say that we have done a lot of work looking at language and cognitive markers and looking at effective treatments, but I really won't have time to get through any of that today. Because what I want to spend the rest of the time talking about is our work looking at developing modeling algorithms to better predict who will respond to treatment and really get to providing personalized treatment recommendations for patients. So I'm going to talk about 3 ongoing projects in this area. The first one is going to build on what I had just talked about. In this case, we're doing machine learning to predict whether somebody is going to end up being a responder to treatment or a non-responder to treatment. Now, the other two projects use behavioral data. They don't include the imaging piece, but one of them is looking at neural networks in bilingual patients, and another one is looking at really large, big data sets to provide therapy outcomes. So I'll talk about all these three ongoing projects in the remaining time I have. So leading to the first topic here, which is using machine learning approaches. As I said, this really builds on our earlier work that I talked to you earlier about. What this work does relative to everything else I presented earlier is take all of those markers, whether it's resting state or lesion volume, into one set of models, and then examine which of these factors are relatively more or less important. Because in all those studies, we were looking at one thing at a time, and this work now brings everything together. So this is a study where we recruited 55 patients with a stroke. They all received different types of treatment, but they all received 12 weeks of intervention. And as I said, we looked at age, education as demographic factors, aphasia quotient, cognitive scores as behavioral factors, and all those imaging metrics I showed you earlier: lesion size, percent spared, FA, resting state, fMRI, connectivity, and so forth. All of these went into the input part of the model. They're all entered in as inputs. And what we were trying to predict was whether somebody could be classified as a responder or a non-responder in the treatment. And so it's important here that here we're looking at percent change at a threshold of 0.25 change. So the responder treatment, they've shown at least a 25% change. We're testing two models here, support vector regression, support vector machine models and random forest models. And in both cases, we're looking for F1 accuracy, precision, and recall that I'll talk to you about. It's important that in this case, we sort of incrementally test all these features by themselves and then put them all together. And we use a leave one out cross-validation approach. What we found here was that support vector SPM models predicted performance pretty well at 90% accuracy. And the top model, the optimal model here, included aphasia severity, demographic data, diffusion-based integrity in white matter tracts, percentage of spared tissue in gray matter regions, and resting state functional connectivity data. Now, it's important to point out that this was not all the features, because the all feature sets, the one that included all the features, was actually not that good. It didn't even just barely touch 80% accuracy. And when we looked at random forest models, they were actually slightly different, but less accurate than SPM because the highest level of accuracy was not yet at 90%. The top model in this case was actually just resting state fMRI, and the all features, when everything was put together, actually didn't do as well. It was not even close to 80% accuracy. The top SPM model correctly classified 51 out of the 55 patients as responders versus non-responders, whereas the random forest model correctly classified 46 out of the 55 patients. So it just wasn't as a strong output, and we can chat about that. What was really interesting about this work, and I think what we're going to pursue at this point, is when we look at the feature set occurrence, when we look at how often each of these features show up in the top combinations of the graph, we see that in both the SVM model and in the random forest model, resting state connectivity is the predominant feature set. They show up in almost all the top combinations of models that best predict treatment outcomes. And so this is work that we think is worth pursuing. But I want to just go back to the earlier part of my talk where I was sort of telling you that it is about the connectivity in the network that's most important, that predicts recovery. And this machine learning work sort of brings it back together, saying, yes, indeed, it is about the way this resting state connectivity predicts outcomes. There's a lot more work to be done here, but at least I think we have something to go chase and keep building down. Now I'm going to switch over to some other work we've been doing lately in bilingualism and bilingual aphasia. And here we've developed a computational simulation using neural networks. And essentially we're trying to simulate what it means to be a bilingual person. So just ignore that, that picture for a second, I'll get to it. What we're doing in this work is we start with a conceptual representation of what it means to be a bilingual person. We have a bilingual semantic map, which is on the top there, and then it's connected to two L1 languages. In this case, we're modeling English and Spanish. So there's an L1 English and an L2 Spanish. The model is called BILEX, and it can simulate representations in the semantic system and L1 and L2 with increasing exposure. So what you see here on the left-hand side is a semantic map that's very well defined in terms of features, semantic attributes. And on the right-hand side, you see an L1 map. In this case, it's English. And again, it's organized by phonetic features. And so the model sort of learns this over time. And what's really important about this work that we've published in the past is that we can simulate a person who's had early L2 age of exposure and early L2 age of acquisition and high exposure as shown by the left-hand quadrant, top left quadrant. It's a very well-defined map. This is somebody who's very comfortable in both languages. But we can also simulate somebody who's had a late L2 age of acquisition and a low exposure in L2. And that you can see in number D. With the ill-defined map. The map is just not as robust. And what you see in this animation is sort of a simulation of how this actually gets represented. On the left-hand side is the semantic map. There's the English phonetic map here and the Spanish phonetic map. In this particular simulation, Spanish is the first language, English is the second language, and you can see as it sort of grows over time, the maps represent themselves very differently. The semantic map is, self-organizing over time, and it ends up having a very rich representation, but the English exposure is very low. So the sort of the representations aren't as rich. And so you can see the time scrolling over it. It's still only 15 years at this point. For the model, and as it keeps going further out, you can see the map gets much more well represented. So what we have done with this work is we've sort of shown that we can simulate a person. We can simulate a bilingual person, understanding the extent to which the maps organize. I don't know if you can see, but the model is still going. And at this point, the English phonetic map is really not well developed. So we can simulate a bilingual person, and we're calling that a pre-stroke model because this is what the map looks like before a person has had a stroke. Then we can simulate aphasia by introducing noise into this model such that it matches the behavioral performance when we're actually testing our patients. So in this particular case, we either introduce a noise in the semantic map and we try to match the performance on the pyramids and palm trees, or we introduce a noise in both the semantic that influences naming, or we introduce damage in all the three maps, in which case you have severe damage in both semantic and naming performance. But the point is we can come up with a post-stroke model where we can match the degree of language impairment in the two languages of a person. We take that data and then we can then simulate treatment outcomes for these people. So what you see down here is the model simulation of what it would be like for this post-stroke model to then receive therapy, quote unquote. And the important thing here is that the model can actually predict treatment outcome, whether the treatment is provided in English in the left-hand case or in Spanish. It doesn't care. The big limitation is that we can only provide therapy in one language. So the model is telling us the outcomes in both languages. And so in this paper that came out in Scientific Reports, we showed that that the model actually does a good job predicting the actual patient data. When it starts off at 2 weeks down here, that it's not very accurate, but by the time therapy is done after 10 weeks, it actually does a very good job of predicting what the patient data would look like. So we're actually got a great treatment simulation model as well. And then based on that, we can actually create a treatment prescription. So what we've done in this work now is we now have the bilingual history, what I showed you in the previous slide, and we can create a pre-stroke model. In this slide, I showed you how we can introduce noise into the model and match the naming and pyramids and palm tree scores. And now we have a post-stroke model. And we just keep creating different ensembles of these models and then put it through a treatment simulation. The key piece here now is once the model goes through so many iterations, it can actually tell us which is the better language to train, because it knows based on its outcomes, which is going to be the language output that's superior. So in this case, it's saying you should probably treat the patient in Spanish and not in English. And so the exciting thing about this is we actually are close to the end of a double-blind randomized control trial when we've actually done this with 48 patients, where we recruit these patients, we do the assessments, we understand their pre-stroke and post-stroke impairment. We then enter that information into the model. The computational models then simulate the outcomes and give us a prescription of what's the right language to train the patient. In this case, the model says train this patient in English. And then in the randomized controlled trial, the patient who's in the experimental group actually gets therapy in English because that's what the model said would be a good language to train. And the patients in the control group get the exact opposite language. And so we're almost done with this study in terms of providing therapy, and we'll be able to come back and validate whether the model actually got it right or wrong. So stay tuned for those results. And the last last project I want to talk about, and then I'll be done, is to talk about our work using big data mining in terms of predicting personalized treatment outcomes. Now, this is work that's very different. Unlike all the work I was telling you about in terms of patient factors, lesion location, and so forth, here we're sort of focusing on that treatment factors. Is what is it about the therapy we can give them? Is it the optimal dosage? Is it the type of treatment, and so forth? And we're trying to see if that predicts treatment outcomes. So it's a different question we're asking here. And we're able to do this because we have access to the Constant Therapy Data Digital Health Platform, which I brought back to BU. This is what the app looks like. If you don't know about it, it's a commercially available app with about 400,000 users, patients, and about 37,000 clinicians. It enables self-managed high-intensity therapy. Patients practice this app at home when they can't go to the therapy sessions. It's evidence-based therapy across 13 skill domains like reading and comprehension and so forth. And patients self-select skills to use to improve on, and then there's a personalized regimen, an algorithm that's automatically adjusted. But I won't get into that. That's just telling you about the details of what's in this app. What we did here was we took data from the app that we had access to. We started with about 238,000 patients. And then we filtered through that to various constraints. And we were only looking for people who were at least a certain, had stroke, had a certain amount of age, and most importantly, practiced this therapy for 10 weeks consecutively. And we ended up with about 2,000 patients here. And just to tell you who they are, they're mostly older adults. Between the ages of 51 and 70, they're between equal males and females, and importantly, they're all, most of them are less than 6 months post-stroke. And not surprisingly, when we looked at the therapy gains in these people, what was sort of obvious to us was that the longer the patients practice treatment, the more the change on something we calculated was the domain score, so how much they improved on any given domain. So the longer they practice, the more they improved. The greater the dosage, so 100,000 hours of total therapy resulted in greater domain score changes, and greater task variety. So mixing things up a little bit also resulted in greater improvements. But more importantly, when we look closer at the optimal therapy dosage, the amount of therapy practice, and we compared people who received 4 weeks of treatment, who practice 4 times a week versus 5 times a week versus 3 times a week, 2 times a week, 1 time a week. What we found was patients who practiced 4 and 5 times a week were associated with significantly greater performance change than patients who practice 1 or 2 days a week. And I just want to point out that the yellow line, which shows 1 day a week, is kind of what standard of care is right now. If you're a stroke patient and you're chronic, you're lucky to get therapy once a week like this. But it's clearly not as good as practicing more often. Obviously, the important point here is that a majority of these individual skill domains show the same sort of trajectory. So it doesn't matter how you slice the pie, you sort of get this more improvement with more practice. So where we're going with this work, now that we have this data, is to really, and we got some recent funding to do this, is to create an online therapy outcomes calculator. What that means is that a patient could go to a website and enter the relevant information in this profile, say, so and so, I've had a stroke, it's less than 6 months. And then they can tell us what domains they want to work on, like reading or memory. And they can also tell us how long they think they can work on it. So can they work on it for 2 months, or they can do therapy for 6 months? And what this data mining we're able to do right now will tell them, or this therapy calculator will tell them the probability of improvement in any skill domain based on this data mining. For instance, if a patient wants to practice reading for about 6 months, the calculator can say there's a high probability that if you start at reading words, you might actually progress to reading paragraphs if you practice this many months, or do the same thing for speaking and so forth. So this is what we're doing this year and next year with this work. So I just want to summarize all the things I had started talking about. Of course, we're really interested in identifying the markers that we can then put into a predictive modeling. But the goal here is to really personalize that rehabilitation based on the 3 or 4 examples I was talking about today. And I do think that there can be a future where if we knew exactly what data to collect in the neural profile and the language and cognitive profile, I think we can get a complete picture that we can then put into these machine learning models, whichever one actually ends up being useful. But it can be used to predict each patient's personal recovery profile trajectory. And based on that information, we'll be able to provide optimal prescriptions on whether the patient should receive intensive therapy or individual therapy or social functional therapy. So I will stop there, and I look forward to questions from the group.